Today we're going to look at how we can mobilize our fat to produce acetyl-CoA, which will feed into the citric acid cycle. So at the end of our last lecture, we were looking at the fat storage in our lipid cells. Our fat cells are adipocytes. And so now we're going to look at how that fat gets mobilized out of the adipocytes. And then once it's transported to your muscle cells or wherever it's going for fuel use, then how you break down those fats. Okay, so first off, let's kind of review a little bit of what we learned last semester. So when I'm talking about fats, of course, I'm primarily talking about things like our fatty acids here. So those would be our fatty acids here would be saturated fatty acids and unsaturated fatty acids. So in both cases, of course, we have our long hydrocarbon chain, and then we've got our carboxylic acid head group. And then you've also got your space filling models down there showing the difference. Remember, our saturated fatty acids pack together very nicely and have high melting points. Our unsaturated fatty acids don't pack together as well and have lower melting points. Now, all of our fats, of course, are usually bound together in triacylglycerides. So again, we have our glycerol, and we're going to bind multiple fatty acids to a single glycerol unit, right? So this would be a triacylglyceride, and then we could cut off individual units to create a diacylglyceride or a monoacylglyceride, right? So we have tags, and we have DAGs, and then monoacylglyceride, depending on how many fatty acid chains you have attached to that glycerol backbone. Okay, so let's take a look at how we mobilize and break those fats out of our fat cells. So all of our fat is going to start stored inside these large lipid droplets, right? So we'll have our triacylglycerols in there. You also have some cholesterol esters in there. So cholesterol, of course, is another one of our lipids, although it's obviously very different from our fatty acids. So they're all stored within these large fat droplets. And you have this protein paralipin here marked in purple, which kind of surrounds the lipid droplet and protects those fats inside of that lipid droplet. So you've got a thin layer of phospholipids and then also proteins coating that as well, which keep all your lipids stored and secured inside this lipid droplet. So our mobilization of fat then is going to be triggered by signals. This would either be glucagon or epinephrine. So those would be our we need energy signals. And these will feed into the adenylyl cyclase, which produces cyclic AMP. So we use that cyclic AMP dependent signaling pathway, and we activate protein kinase A. So we've seen this kinase before, of course, when we're regulating glycolysis and gluconeogenesis. Once we activate protein kinase A, that's going to activate and phosphorylate a couple of different components. One is our hormone-sensitive lipase. So our lipase is going to take those triacylglycerols and diacylglycerols, and we're going to start to cut them down into eventually our fatty acids. And we're going to export those fats as fatty acids to the rest of the body. So we phosphorylate the hormone-sensitive lipase, or HSL, to activate that. And then we also phosphorylate paralipin. And the phosphorylation of paralipin is going to basically decrease that protection around our lipid droplet so that our lipases can access the lipid droplet and begin to break apart those lipids. It also releases this other protein over here, which is normally bound to paralipin. So CGI58 will also activate lipases to start cutting up our lipids. There's actually several different types of lipases that are active here. So this is a triacylglycerol lipase, TGL, which cuts off our first fatty acid from the triacylglycerol. So we go down to just 2 fatty acids and 1 free fatty acid. The hormone-sensitive lipase cuts the diacylglycerol down to the monoacylglycerol. And then we have our monoacylglycerol lipase down here that cuts off that last fatty acid. So in the end, we get our glycerol, which is the backbone of all of our triacylglycerides, and then we also release those 3 fatty acids. Once we have the fatty acids released, then those will go into our bloodstream. Serum albumin here is used to carry those fatty acids, since of course our fatty acids are all nonpolar and can't transport through the bloodstream on their own. So serum albumin will carry those fatty acids, and then we can transport them into the cells where they're needed for energy. So in this case, they're entering a muscle cell, and then they're going to go through oxidation before they enter the citric acid cycle. Once we break apart that lipid and we get it into the cells that need it, now we have to break it down to produce our energy. Of course, we have 2 different components that we can break apart for energy, right? We have our glycerol, which was the backbone of our triacylglycerides. And then we also have our fatty acid chains. Glycerol actually enables us to make some ATP without using the citric acid cycle. So you can get some anaerobic production of energy from fats, but the majority of our fats, all the energy comes from the fatty acids, where you would be breaking it down to acetyl-CoA, which has to go through the citric acid cycle. And again, if you're going through the citric acid cycle, you're dependent on your aerobic conditions. You have to have oxygen. We start out with our glycerol. So glycerol, we've got our 3 carbons, each with an alcohol functional group. So these would be our attachment points here, where we used to have our free fatty acids. After our lipases cut them off, we just have our normal alcohol functional groups from the glycerol. So our first step is basically going to be a priming step. We're going to use the glycerol kinase to transfer a phosphate group onto our glycerol. So we're making glycerol, L-glycerol 3-phosphate. So once we add our phosphate at the 3rd position, so we attach it to our hydroxyl group at the 3rd position, and that now makes our glycerol chiral. So initially we're not chiral because we're asymmetrical. Now we're chiral, and we're specifically in the L enantiomer. So this reaction will cost us 1 ATP, Again, very similar to our preparatory phase in glycolysis. Now we're going to take our phosphorylated glycerol. And of course, one thing you should notice with glycerol is we have a 3-carbon molecule. We already, of course, attached our phosphate. And so if we do an oxidation step, we can convert it to dihydroxyacetone phosphate. We've got the phosphate group at one end. We've got our alcohol at the other end. And all we have to do is convert our central alcohol into a ketone to get to our dihydroxyacetone phosphate. So this is through our glycerol 3-phosphate dehydrogenase. So we're doing an oxidation step. So again, we have a dehydrogenase as our enzyme. And again, we're just using the name of our substrate as in the enzyme name. Here we get our first energy production from this pathway. So we produce NADH, which of course can be used under aerobic conditions for energy. This will feed into our eventual oxidative phosphorylation. And now with our dihydroxyacetone phosphate, we have a glycolysis intermediate, right? So this then can just be fed into our normal glycolysis pathway. We take that dihydroxyacetone phosphate, and we can just feed it into the triosphosphate isomerase to produce our normal glyceraldehyde 3-phosphate. So this would then enter glycolysis. We have the glyceraldehyde 3-phosphate already, so we're already halfway through the pathway. And so then we can convert this to get our, this would be a 4 ATP yield from that glyceraldehyde 3-phosphate. So we lost, or actually just 2 ATP rather, because we only have 1 unit of our glyceraldehyde 3-phosphate. So we lost 1 ATP initially with our glycerol kinase. We gain 2 ATP once that glyceraldehyde 3-phosphate goes through our full glycolysis pathway. And then we also had our NADH, which under aerobic conditions will give us the 2.5 ATP. So the glycerol allows our fats to feed somewhat into glycolysis. Again, this is really very small amounts of energy, right? We get just a couple ATP units. We get a little bit of ATP through NADH also, but it's not really any major energy production. Most of that energy is going to come through our through the fatty acid chains, through glycolysis. But I mean, you would actually get more than that in the end, because right, glycolysis would just feed us into eventually acetyl-CoA, right? And each acetyl-CoA unit then will produce more energy from the citric acid cycle, right? So for every acetyl-CoA that we produce, this goes into the citric acid cycle. We get 1 GTP, and then we get our 3 NADH, and we get our 1 FADH2. Actually, I guess we would get, we also get our 1 extra NADH, or 2 extra NADH in the glycolysis pathway as well. So yeah, we'd get some more from the glycolysis, more NADH, and then the citric acid cycle itself gives us basically 1 plus 3 times 2.5, so that's 7.5, plus 1.5 would be 9, plus 1 is 10, right? So in the citric acid cycle, we get 10 ATP per acetyl-CoA. And then we also get a couple more NADH as we go from glyceraldehyde 3-phosphate to that acetyl-CoA. So you would get a little bit more than that when you count what happens in the citric acid cycle as well. And again, that will allow us to get some energy even if we don't use the citric acid cycle at all, right? So you would get at least 1 ATP. So you lose 1 ATP from the glycerol kinase, and then you produce 2 ATP from the glyceraldehyde 3-phosphate going to pyruvate. And then you could convert that pyruvate to lactate, regenerate your NADH, and you could continue doing that indefinitely without using any oxygen. Okay, so that gives us a little bit of energy production. Now, for most of our energy production, we're going to take our fatty acids and we're going to cut them up to make our individual acetyl-CoA units. So we're going to take our fatty acids, so that's represented here. We have our carboxylic acid at the end of our fatty acid. Your R group here represents your fatty acid chain. This is going to vary in length. Most common is 16, but well, I guess it'd be 15. It's 16 carbons total, counting the carboxylic acid. But of course, we can have a wide range of different sizes for these fatty acids. In order to convert the fatty acids to acetyl-CoA, the first thing we have to do is make a fatty acyl-CoA. So we're going to take our fatty acid and we're going to attach our CoA, coenzyme A, to make this fatty acyl-CoA. So that's going to occur through these 2 main steps here. This is catalyzed by a series of fatty acyl-CoA synthetase enzymes. There are a couple different isozymes of this enzyme that work on different sized fatty acids. Right, so you might have a very long fatty acid with lots of carbons. You can have fairly short fatty acids with only a few carbons, and you'll have different isozymes of the same synthetase enzyme that will catalyze this reaction for those different sized fatty acids. Because our thioester here within the fatty acyl-CoA is a relatively high energy bond, we have to use actually 2 phosphates in order to form that bond. So we're going to use 1 ATP. We're going to do a nucleophilic attack. We release 2 of our phosphates as our pyrophosphate. We temporarily attach that adenosine with our phosphate. So we adenylate our fatty acid. And then we do another nucleophilic attack from our coenzyme A to produce that fatty acyl-CoA. Our pyrophosphate gets broken apart again by our pyrophosphatase. And so altogether we have a very favorable reaction. So we get our favorable pyrophosphate hydrolysis, and our this step is favorable overall as well, as we're breaking off those 2 units of phosphate. So we're going from ATP to AMP, so effectively we're costing 2 ATP units per our fatty acid. So once we have those fatty acyl-CoAs, so all of that stuff can occur in our cytoplasm, right? So we break down our fats into fatty acids, we break down that glycerol and feed it into glycolysis. But now in order to break down the rest of our fatty acid, we're actually going to move it into our mitochondria. So this is going to be our beta oxidation, which will occur inside our mitochondria. So first we have to, of course, transport inside the mitochondria, because our mitochondria has its own membrane that blocks transport of molecules across it. For very small fatty acids, we can actually, they can just move right through the membrane. So the membrane, of course, has a nonpolar core. And so if you have something that's small and relatively hydrophobic, it can move through the membrane fairly easily. So small fatty acids can just move right through. But for any of our larger fatty acids, we have to use this acyl-carnitine transporter. So this is our carnitine molecule right here. And we're going to temporarily attach our fatty acid to carnitine. It's going to be attached to this hydroxyl group right here. And that's going to allow us to then transport the fatty acid into our mitochondria. So we temporarily attach here and then transport that using a specific membrane transporter into the mitochondria. So here's what that transport process looks like. So we start out with our acyl-CoA that we just synthesized in the cytoplasm. We take that fatty acyl-CoA and we're going to use a carnitine acyltransferase enzyme, which will take our fatty acid chain. So here's our fatty acid. It's attached onto our coenzyme A. We release our coenzyme A and transport that fatty acid onto our carnitine instead. So we transfer from coenzyme A onto the carnitine, and then at that point, it can be transported through the membrane of the mitochondria using that acyl-carnitine co-transporter. So this transporter converts 1 unit of the acyl-carnitine in, and it transports 1 unit of carnitine out at the same time. So our carnitine is basically following a cycle where it goes in, to the membrane or into the mitochondria, carrying that fatty acid. It's then going to release that fatty acid using a second carnitine acetyltransferase. And then the free carnitine is going to get transported back out to the cytosol, where it will pick up another unit of fatty acid. So our second carnitine acetyltransferase is going to take our free coenzyme A and transfer our fatty acid onto that coenzyme A to again produce our fatty acyl-CoA. So overall, nothing is used or consumed in this pathway. We're just shuttling stuff back and forth, right? We have our fatty acyl-CoA outside in the cytosol. We release coenzyme A and put it onto our carnitine to make that acylcarnitine. We transport the acylcarnitine in. We release it, put that fatty acid onto our coenzyme A again inside the mitochondria, and then feed our carnitine back out to the cytosol to pick up more fatty acids. So carnitine actually is something that I believe some athletes take as a supplement sometimes, because it's thought that, yeah, by having extra carnitine, you might be able to transport more fatty acids into the mitochondria. But since you're not necessarily using carnitine up in this process, right? It's not like we really consume that carnitine. So biochemically speaking, I'm not sure how useful it would actually be to have a larger amount of carnitine within this transport mechanism. Okay, so now we have our fatty acyl-CoA inside our mitochondria, and we're going to carry out our what we call a beta oxidation in order to produce our acetyl-CoA units. So we're going to start out from our fatty acid. We're going to oxidize it, which is going to produce our acetyl-CoA, and then we'll feed that into the citric acid cycle. Remember, the citric acid cycle also occurs inside the mitochondria as well. So we'll be able to immediately produce that acetyl-CoA and feed it directly into our citric acid cycle. The citric acid cycle, again, is going to be feeding our electrons, which will be carried by NADH and FADH, into the oxidative phosphorylation, where we'll actually be producing our ATP. So for any fatty acid, we're basically going to cut it off 2 carbons at a time. So you can see in the diagram here, it's marked off these blue lines. We're cutting off 2 units at a time to make those acetyl-CoA units. So for a 16 carbon fat, you get 8 acetyl-CoA units, which will then produce our energy in the citric acid cycle. So as we go through the beta oxidation pathway, each time we go through, we're going to be cutting off a single acetyl-CoA unit. So we'll basically cut off 2 units, 2 carbons as 1 acetyl-CoA, and then of course we have to oxidize the next 2 carbons to get them to our same thioester functional group, and then we'll cut it off again. Right, so our 16 carbon palmitoyl-CoA gets cut off in 7 cycles to produce our 8 total acetyl-CoA. Let's look at how that happens. So we start off with our palmitoyl-CoA, so our fatty acyl-CoA. This could be any fatty acid. It doesn't have to be palmitoyl. So this can just be generically referred to as an acyl-CoA. Palmitoyl is the 16 carbon, which of course, again, is the most common, but yeah, it could be anyone. So at the end, we of course have our thioester attached to our coenzyme A. And beyond the thioester, we just have carbons stretching back. So we've got our long carbon chain. Extending for 16 carbons in the case of our palmitoyl-CoA. So the first step, we're going to use dehydrogenase. So our goal is to oxidize this so that we have a new thioester as soon as we remove our first two carbons. Right, so we're going to remove our first two carbons by breaking this bond here. But that means we have to oxidize this carbon here so that we can form a new thioester to make our next unit of acetyl-CoA. So we already have one unit of acetyl-CoA ready to go, but we have to oxidize to get the next unit ready. So our first step is that dehydrogenase. So we're going to form an alkene from our alkane. So our alkene is getting oxidized to the alkene. So we still have our coenzyme A at the end, but now we're going to add a alkene double bond. So we add our alkene bond. It's a trans bond there. It's trans delta 2 enoyl-CoA. So if you remember, the delta sign was counting how many carbons it was from the start of our chain. So we have, this would be carbon number 1, this would be carbon number 2. The double bond is starting at the second carbon. Right, so it tells us it's at the second carbon. It also tells us it's trans. Of course, the EN here indicates that there's an alkene, and then we have our original oil-CoA indicating that fatty acid attached to our coenzyme A. We're again using FEDH here. So remember, we used FEDH in the citric acid cycle also when we did our succinate dehydrogenase, which converted from succinate to the fumarate that has the double bond. So this is a very common event that we see. Whenever we go from a alkane to an alkene in the body, we're usually using FEDH. So after we oxidize it, the next step, we're going to do a hydration reaction with our alkene double bond. So we're going to take that double bond and we add water to it in a hydration reaction. So our water comes in. We have our same thioester. And now we have our hydroxy group added as we eliminate that alkene double bond. So we add our water to replace that double bond. And this becomes a L-beta hydroxy acyl-CoA. So again, remember our nomenclature, we use alpha to refer to the site next to our carbonyl, and beta would refer to the next site after that. So our hydroxy group is at the beta position relative to our thioester carbonyl. We still have the same acyl-CoA overall, right? It's the same lipid that we had in the first step. It's just that we added that hydroxy group right here. So the names really indicate exactly what we're looking at in terms of the structure. So now we have our alcohol here. So you've got our thioester, our alcohol, 2 carbons away from the thioester. So that L-beta hydroxyacyl-CoA. Now we use another dehydrogenase to oxidize that alcohol. So we're going to produce NADH this time. We get NADH, of course, when we're oxidizing our alcohol here to a ketone. When we did the alkane to the alkene, we just produced the FADH. So we still have our thioester, but now we have our ketone. So it's a beta ketoacyl-CoA. Again, this would be our alpha site, and this is our beta. And we have that ketone at that beta position. So now at this point, we have our 2 carbons over here that make up a single unit of acetyl-CoA. And our next carbon has been oxidized to a carbonyl, right? So the only thing we need to add is another unit of coenzyme A, and that can attach here and make our thioester. So this step is going to use an acyl-CoA acetyltransferase, or a thiolase, which is going to cut off our single acetyl-CoA unit. All right, so this will release our acetyl-CoA and attach that new acetyl-CoA right there by the bond that we're breaking. So we end up forming a acyl-CoA, which looks identical to what we had originally. The only difference is that we've cut off 2 carbons, right? So instead of our palmitoyl-CoA that we had initially, in this case, we would have myristoyl-CoA, which is our 14-carbon fatty acid. And this process would continue, right? So we could keep going through the same steps, oxidizing. So we oxidize, we hydrate, we oxidize, and we cut it off. 4 basic steps, each time just reducing that length by 2. So here's kind of the look at the overall pathway through those steps. The electrons that we're getting out of it are being fed, of course, into that respiratory chain and oxidative phosphorylation. So we're getting those electrons are actually going to be fed directly away from our NADH and FADH2, so we can regenerate our NAD and FAD to continue oxidizing. So we're going to kind of immediately transfer those into the oxidative phosphorylation. Now, one thing you can notice here is that, again, the overall pathway is very similar to what we see in the citric acid cycle, right? We start out with our alkane to alkene. So that we start off with our acyl-CoA. We oxidize it to the alkene, right? So this was our acyl-CoA dehydrogenase, or in the case of the citric acid cycle, this would be our succinate dehydrogenase. You then take that alkene and you hydrate it. This gives you your alcohol. So this was our enoyl-CoA hydratase in the case of our fatty acid. In the case of the citric acid cycle, this would be our fumarate here, right? We're taking fumarate and converting it to our malate. And then, again, our final step, we oxidize from that alcohol to the ketone. This is done by our beta-hydroxy dehydrogenase, beta-hydroxyacyl hydrogenase, versus, of course, in the case of the citric acid cycle, we had our malate dehydrogenase that converted from the alcohol to the ketone from malate to oxaloacetate. So it's a very similar pattern that's kind of followed this overall oxidative process. Alkene to alkene, alkene to alcohol, alcohol to ketone. And again, in our case here, right, we can repeat that continually, right? Because we just cut off that ketone and we get a new alkane, and you just keep going through that cycle. We also see a very similar pathway with amino acids as well, which we'll look at this more later. For amino acids, this is in the oxidation of isoleucine. You again see that same general pathway. You start with an alkane, oxidize it to the alkene, hydrate it to the alcohol, and then oxidize it to the ketone. Each time you're producing FADH and NADH. The oxidation pathway is actually catalyzed by kind of a single enzyme complex. So it's individual enzyme functions, but they're often actually together within kind of a single complex, similar to our pyruvate dehydrogenase. So we've got multiple different enzymes all bound together in this larger complex. It does vary quite a bit depending on what system you're looking at, and but yeah, they're often together in one complex rather than in individual components. So you do see some differences. So gram-positive bacteria here, you do actually have some individual enzymes that are separate, and you then are exporting intermediates between those individual enzymes. Gram-negative bacteria tend to have them more clumped together in a single product. For mitochondria and eukaryotes, you tend to have them more clustered. Here they're actually clustered in the membrane, and you're just moving from one enzyme to another as you're going through those 4 steps. So you're not really, it's not like you go from one enzyme to the next, but you're kind of all within one complex, and you're just moving around within that complex to catalyze those 4 individual steps. So if we're looking at, again, our palmitic acid, which would become the palmitoyl-CoA, we have our 16 carbons. So because each of our acetyl-CoA are 2 carbons, we get 8 molecules of acetyl-CoA. Each time we go through the cycle, we form 1 FADH when we go from the acyl-CoA to the trans delta-2 enoyl-CoA. And we also form 1 NADH when we go from the beta-hydroxy acyl-CoA to our beta keto acyl-CoA. So we'll go through the cycle only 7 times because once we get to the end, you'll have one cycle where you produce 2 acetyl-CoA, right? So once we're down to 4 carbons, when we get down to our last 4 carbons, and then we oxidize it all the way up to our beta keto acyl-CoA. When we cut this with our thiolase, we just get 2 units of acetyl-CoA. Right, so this would just go to two units of acetyl-CoA, and then we don't have to continue our cycle. So we only need 7 cycles, and we get 2 acetyl-CoA in our final cycle. And then we also, each unit of acetyl-CoA will go into our citric acid cycle. Again, as I mentioned earlier, that would produce 1 GTP, 3 NADH, and 1 FADH2. And then all of our electrons from each of these will go into the electron transport chain where we do our oxidative phosphorylation. So in terms of energy production then, we have, so we're producing 7 FAD and 7 NAD from the breakdown of palmitic acid or palmitoyl-CoA. Right, so that'd be 7 times 1.5. So that gets us our 10.5 ATP here. 7 times 2.5 gets us 17.5 ATP here from the NADH. So that's from the 7 cycles of our palmitic acid going through the beta oxidation. We then take our 8 units of acetyl-CoA, and we're going to run the citric acid cycle 8 times, which each of those 8 units. So we started out with 16 carbons, which led to 7 oxidation cycles, which produced 8 acetyl-CoA. For every acetyl-CoA that's going through the citric acid cycle, we again get our 3 NADH. So 8 times 3. And we also get our 1 GTP here from the succinyl-CoA synthetase. And our 8 FADH from the 8 times going through the succinate dehydrogenase. So again, each of these we can multiply by 2.5. Each of our FADH we multiply by 1.5. And we get our corresponding numbers of ATP that form. If we add all of these up, you get 108 ATP for the total energy produced from 1 unit of palmitoyl-CoA. So if you compare that to glucose, remember glucose produces 30 to 32 ATP. So you have 30 ATP for 1 unit of glucose versus 108 for 1 unit of palmitoyl-CoA. Or in terms of ATP per carbon, That would be 5 ATP per carbon for glucose. As opposed to for our palmitoyl-CoA, it would be 6.75 ATP per carbon. So that's, again, that advantage that we get because our fatty acid carbons are more reduced initially. So you can produce more ATP per carbon initially. We would lose, of course, some here as well when you're, because of the initial conversion to the palmitoyl-CoA. So if we're looking at the energy from palmitic acid, so starting from our fatty acid and going all the way to our energy production, here you would only get out 106 ATP from a single unit of palmitic acid, right? Because you lose 2 ATP from the acyl-CoA synthetase. So that slightly reduces the efficiency of it, but overall, we are producing more energy per carbon than we do for glucose. So essentially, for every 2 carbons we have, we're getting out 4 ATP in the beta oxidation. So this is giving us 4 ATP per 2 carbon unit. And then when we feed that into the citric acid cycle, we get 10 ATP per 2 carbon unit. Now, of course, that's the 4 ATP per 2 carbons isn't strictly true because we only do, we only run through the cycle for the number of carbons, 2 carbon units minus 1, right? So we don't get the extra 4 ATP from our last cycle because we already have the, we get that 2 acetyl-CoA from our last time through the cycle. Okay, so this shows us the overall energy production pathway from our fat. So starting from that fat, producing our acyl-CoA, and then oxidizing that to get our acetyl-CoA. So you can see that the metabolic pathway and also the large amount of energy that we produce from that pathway. So we get our large 108 ATP from that palmitoyl-CoA. Then we'll stop here for today. Next week, we'll look at how we change up the oxidation pathway if we have more complicated fatty acids. So here, of course, we looked at a even-numbered fatty acid, which is convenient because you can easily break it into our 2-carbon acetyl-CoA units. Odd-numbered fatty acids have to be treated a little bit differently because you can't just break it down into 2 carbon units. You're always going to have one, either a single carbon or a 3-carbon unit at the end. And then I'll also show you how we deal with unsaturated fatty acids because, again, if you have a double bond already present, that's going to mess up your normal beta oxidation pathway. So I have slightly different alternate pathways that we'll use for those special cases.